Assalamu alaikum. So let's do DNA sequencing now. So what is DNA sequencing? Let's define it first. So DNA sequencing is basically the process of knowing the exact order of nucleotides in a genome, in a piece of DNA. It can be the human genome, for example, or it can be a small gene. It can be a VNTR, it can be STR, it can be whatever it is, telomere, you name it. So basically, it's knowing the exact order of nucleotides in a piece of DNA. So it's like taking a book and saying, well, the order of letters is T H E A P P L E I S R E D and so on. So the thing is, it's meaningless, right? I mean, it's just letters scattered one after, well, not scattered, but ordered one after the other. But then by knowing this order, we can tell a lot. We can identify genes and their localization. We can say that here's a gene, here is a VNTR, here is an STR. Okay, and we would know where they are localized on chromosome 1, the short arm, the long arm, chromosome 2, near the telomere, and so on. We can identify protein structure and function. So after we know that here we have a particular gene, we would be able to translate this order of nucleotides into amino acids, as we will learn later on in translation. We can identify DNA mutations. So we compare the sequence of DNA of an individual compared to a healthy person. And we would be able to pinpoint, to identify, to assign a certain position in the chromosome, in the DNA, and say, here's a mutation that can be pathogenic, that can cause the disease. So we would be able to know genetic variations among individuals in health and disease. A certain population, for example, we say that um, people who have a certain disease in this population are more likely to have A in this position. Now, what does that mean? We may not know, but at least we know that this A is linked to having a disease, like we talked about before in single nucleotide polymorphisms. We can predict disease susceptibility and treatment efficiency. We would be able to say, you need to take this dose of this medication because you have a TTT, okay, in that position. Or we say that be careful, in 20 years of time, you might have a 70% chance of having a certain disease. Or we would know evolutionary conservation. We would know how things evolved in life by comparing organisms and species to each other. So we can learn a lot. Now, this is little history. The DNA was sequenced for viruses and prokaryotes first because their genomes are simple. And then human mitochondrial DNA was done later on. That was like in the 70s and 80s. And then the first eukaryotic genome was sequenced, specifically a yeast, a species known as Saccharomyces cerevisiae. The genome of multicellular organisms were done, specifically C. elegans, then Drosophila, and the Human Genome Project started in 1990. And like I said before, it was assumed that it was estimated that it would cost like something like $3 billion to sequence the 3 billion nucleotides in the human genome, and that it would take 15 years. It was done in 2004. The first draft was actually published in 2004, but it was completed, and 90% of the human genome was done at that time, and it was completed just last year in August 2023. Now, why did it take 20 years to complete that 10% of the human genome? Because of the repetitive sequences in the centromeres and telomeres and other regions as well, including the Y chromosome. The Y chromosome was problematic. So many repetitive DNA sequences. So that's a bit of a history. Now, the major findings of the Human Genome Project and other initiatives such as the ENCODE project revealed that the number of protein coding genes is less than 20,000. Okay. With less than 20, this is amazing because we're talking about the same number as the number of genes in mice. Many of the human genes are common with other species like yeast, Drosophila, C. elegans. Like I said, it's the same number of genes as in mice. But the Human Genome Project also revealed that the number of regulatory elements is really significant. It's about 30% of the human genome. And that indicates that regulation is really important in making a human being. Something else is that the 20,000 or so gene coding proteins cover only 2% of the human, actually less than 2% of the human genome, but the number of regions, DNA regions, or the portion of the human DNA that is transcribed is about 75%. And most of the human DNA makes non-coding RNA molecules, RNA that is transcribed but not translated, including microRNA, long non-coding RNA, and so on. And they seem to be relevant. I'll show you how relevant they are, or I'll show you a sign of how relevant they are. Now, this is not for you to memorize, but just showing you the genome size, protein coding genes, number of chromosomes in different organisms like E. coli, for example. It's about 4.6 million base pairs, encoding 4,300 protein coding genes, and there's only one chromosome. Things get more and more complex as organisms become more and more complex. So mouse genome is very sort of similar to human genome, and the number of genes, protein coding genes, is also similar as well. Now, this is viruses, this is organelles, mitochondrial DNA, for example, dogs, chimpanzees. So compare chimpanzees' genome to human genome, really same thing as protein coding genes and so on. So really things are quite similar. Again, this is not for you to memorize, just to show you the sizes of the genomes in different organisms, like in bacteria, in prokaryotes. This is E. coli is right here, and this is mammals, including humans. Okay, note that the size of the genome of the human genome is less than in amphibians or in certain fish, which is quite amazing, or even in certain plants. Okay, or protists. I mean, primitive organisms. So things are quite heterogeneous, and genomes can be complex as well. So this is what I want to show you. So if we look at protein coding genes, we see that there's similarity among different organisms, primitive as well as complex, such as humans and mice. So the number of genes really is sort of similar, if you want to look at it this way. Well, what's really important is the non-coding genes, like microRNA. So genes that are transcribed into microRNA, but these microRNA molecules are non-coding, meaning that they are not translated into proteins. So the number of microRNA encoding genes is really large. in humans and mice compared to primitive organisms, such as Drosophila, which is fruit fly, C. elegans, the worm that I keep talking about. Yeast doesn't have anything, no microRNA whatsoever. Look at the long non-coding RNA. The more there's a correlation between complexity and the number of long non-coding RNA, and that gives us a sign, a signal, a mark that these non-coding RNA molecules seem to play a very important role in human cells and mice as well. So what do they do? We know little, we know some, but not everything, and I'll talk about the functions of these RNA molecules later on. So let's get into sequencing. So sequencing the DNA depends on the use of a special molecule. But before, when DNA is synthesized, the enzyme is known as DNA polymerase, and the substrate is known as a deoxyribonucleoside triphosphate, 3 phosphate groups. And when the nucleotide is added, we have they're added at the 3' end of the sugar molecule, at the hydroxyl group right here. So, and that involves the release of 2 phosphate groups to provide energy for that reaction, for the polymerization reaction. Okay, so remember, we're talking about a phosphodiester bond between the phosphate that is linked to carbon number 5, right here, this one here, with carbon number 3, with the hydroxyl group of carbon 3. So that's important. Great, and release of energy. Now, so what's that special molecule? So what's the special molecule? Well, it's known as a dideoxyribonucleoside triphosphate. Now, the idea here is that this is the normal substrate of DNA polymerase. Deoxyribonucleoside triphosphate, we have a hydroxyl group right here, and this is where the phosphodiester bond is formed. And we have a deoxy sugar because we have an H, we don't have an OH right here. Okay, great. Now, the dideoxy, not only that it has a deoxygenated carbon right here, it also has a deoxygenated carbon right here as well. This means that this substrate, when added to DNA, it cannot form a phosphodiester bond. And we say that synthesis of DNA is terminated. It stops when this molecule is inserted into the DNA. So that's the whole purpose of the dideoxy triphosphate. So let's get into the methodology. You can read the slide on your own. Okay, I'm not gonna read it for you, as well as this slide right here. But that's what we do. So this is called, by the way, the Sanger sequencing method, because the scientist who came up with it is known as something Sanger. So it's known as the Sanger sequencing method, and it depends on the use of the dideoxyribonucleoside triphosphate. So here's the idea. We're going to have 4 tubes. In these 4 tubes, we're going to have the DNA template that we want to sequence. So that's the DNA template. Of course, it's double-stranded, but we separate the 2 strands from each other. We denature the DNA eventually during the reaction, so we have a single-stranded DNA molecule. This is the DNA that we want to sequence. We want to identify what the sequence of this thing is. So that's one. We add the DNA template. We add the DNA polymerase. The DNA polymerase needs 2 things, like I said before. Substrate, just like any other enzyme, substrates, the 4 substrates. And the 4 substrates that are added are deoxy-ATP, deoxy-GTP, deoxy-CTP, deoxy-TTP. Great. It also, DNA polymerase requires a primer because it cannot synthesize DNA de novo. It cannot synthesize it from scratch. It needs something to start at. So a primer is added, okay, and we know what the sequence of the primer is. So we have to have a little idea about the sequence of the DNA template. Now, sometimes we don't know what the sequence is. Sometimes we know what it is, but we just want to make sure that there is no mutation, there is no change in the DNA sequence, and that's why we're sequencing the DNA. But we have to have an idea about the sequence of the template that we want to sequence so that we can use an appropriate primer for this DNA template. And then we add very little dideoxy substrate, dideoxynucleotide. So this is added at low concentration. So we have 4 tubes. Each tube has DNA template, a primer, just one primer, okay, substrates, DNA polymerase, and then to tube number 1, we add dideoxy-GTP. To tube number 2, we add dideoxy-CTP. Tube number 3, we add dideoxy-TTP. Tube number 4, we add dideoxy-ATP at low concentrations. So the DNA polymerase starts to synthesize the DNA, the complementary strand of the template. So let's say the primer ends with a C. And this is the tube that contains the dideoxyribonucleotide. So it sees, here's a C, it sees a T, it has an A. It sees a C, it has a G. Great. Now, it has 2 options. The DNA polymerase has 2 options. Either it adds a deoxy G or it adds a dideoxy G. Most probably, it's going to add deoxy G. Why? Because the concentration of dideoxy G is low. So most probably, it's going to add a normal G, deoxy G. And when it adds a normal G, synthesis is going to continue. But for some DNA molecules, it's going to add a dideoxy G. And as a result, synthesis stops for this fragment right here. Now, what I forgot to tell you, and I'm sorry about that, is that the primer is labeled with radioactivity. So it gives a signal. Now, and let's say that we have 1,000 molecules of this template. Remember, I'm not talking about one molecule of template, one molecule of DNA polymerase. I'm talking about large numbers of molecules of everything. Of the template, primers, enzymes, substrates, and dideoxy G, except that the dideoxy G is added at lower concentration than the regular substrates. So let's say that we have 1,000 templates having primers hybridized to them. The DNA polymerase is going to add, let's say, a deoxy G, normal G, to 900 of those. And it's going to add to 100 of the 1,000 the dideoxy G. So for those 100 with the dideoxy G, synthesis stops. For the other 900, synthesis continues. So it sees an A, it has a T, it has another A, T, another a G, it has a C, A, T, and then it hits a C. And again, there are 2 options. Either the DNA polymerase adds a deoxy G or it adds a dideoxy G. So let's say, since deoxy G is more abundant than the dideoxy G, the dideoxy G is added to 100 of those 900 molecules. And for the other 800, a deoxy G is added. So for those 800 molecules, synthesis continues. And for the 100 where we have a dideoxy G, synthesis of the DNA stops, and so on. So for the other, what do you say? 800 synthesis continues, and again there's a C. So let's say that it adds a dideoxy G to 100 and a deoxy G to the other 700, and so on. So here for the strands where the dideoxy G is added, synthesis stops. It's terminated. And that's the whole purpose of the dideoxy. Okay, and then what? Remember that in the other tubes where we have, let's say, a tube that contains a dideoxy T in addition to all four substrates, whenever there's an A, synthesis will stop when the dideoxy T is added. Okay, and same thing for the other two tubes. Okay, so here we have double-stranded DNA and we want to sequence, let's say, this strand right here. We add a primer. We separate the two strands and we add a primer. The primer is labeled. Okay, so that's labeled primer. The primer is labeled. We add the four substrates. Okay, and then to tube number 1, we add dideoxy A, tube number 2, dideoxy T, dideoxy C, dideoxy G. Okay, so we have 4 tubes right here. And we add the DNA polymerase. Now the DNA polymerase is going to start synthesizing DNA. So there is a T right here. It's going to add an A. Now in this tube, it's going to add a normal A, a deoxy A, or a dideoxy A. If it adds a dideoxy A, then synthesis stops here. If it adds a deoxy A, synthesis continues until it hits another T, and then there are two options: dideoxy A or deoxy A, and so on. So we're going to get in each tube, we're going to get fragments that are different in sizes. Remember that the primers are labeled. Now, same thing here in this tube that contains dideoxy T. We're going to get fragments of different lengths. Same thing here, same thing there. Now, look at this one right here. This one differs from that one by only one nucleotide. Here we have just an A, and here we have A and T. Look at this one here. This one is larger by one nucleotide as well. So we take these 4 tubes and we put them on a gel, a special gel that has high resolution, meaning that it can separate DNA fragments that differ by one nucleotide. Amazing resolution. Great. So here we add the reaction for the tube that contains the dideoxy A. This one here contains the reaction for the dideoxy T, dideoxy C, dideoxy G, and so on. And the fragments are separated, and they're separated based on size, except that we can differentiate between two fragments that differ by one nucleotide. Great. So this is the so migration movement of the strands goes downward. Okay, so the smallest, the shortest DNA fragments move really fast compared to the larger ones. So here we have the large DNA fragments, here we have the small DNA fragments. This is the shortest. So we know that the shortest contains or is belongs to the reaction that where we have a dideoxy A added to it. So we have an A right here. Remember, synthesis goes from 5' to 3'. Okay, so when A is added, it's near the 5' end. So here we have an A, and then we have a T, and then we have a G. Okay, so they differ by one nucleotide. Okay, now we have a T, right? We have a C, we have an A, we have a G. So that's really the sequence. So the 5' end starts right here. So we say that the sequence of the DNA that is synthesized is A, T, G, T, C, A, G, and so on. So here we have the 5' end, and here we have the 3' end of the newly synthesized DNA. Now we know what the sequence of the template is because it's complementary, right? It's complementary to this sequence right here. So now we know what the sequence is. Now look at these examples. So here we have the 4 reactions, dideoxy-A, T, C, G, and so on. So what is the sequence of the newly synthesized DNA? Well, you say it's T, G, C, G, 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 C, TT, then A, and so on. So you would be able to know what the sequence of the newly synthesized DNA is. And of course, by flipping the letters, so you have a T, you have a sorry, right here, instead of A you have a T, instead of T you have an A, and so on. You would be able to know what the sequence of the complementary strand is. Remember that DNA, double-stranded DNA, is anti-parallel. So if this strand, you start right here, 5' to 3', for the complementary strand, you have to go 5' to 3' the opposite way, the opposite direction. So keep that in mind. Okay, again, we have here the DNA labeled with radioactivity. So let's look at this example again. Let's say that we have 2 fragments that line up at the same level. What does that mean? It means that we have fragments, 2 fragments that have the same length, and that's why they migrate, they move through the gel at the same speed, and that's why they line up at the same level in the gel. What does that mean? Remember that we are diploid. We have 2 copies of every chromosome, of every piece of DNA. We take one from the mother, we take the other from the father. So if I take a chromosome from my mother and it has a sequence and the same thing, and I look at the same sequence at the chromosome that I got from my father, I would expect that there would be a difference. It could be a single nucleotide polymorphism, a G in the maternal chromosome and A in the paternal chromosome. So at that particular location, so it's going to be A, T, T, G, G, C, C, A, okay, and so on and so on. And then we get to a certain point, there's an A in here, there is a C on the other chromosome. So this means that the person is, we say, heterozygous for this particular position. That's one. Now, let's look at this example right here. We expect that there is a band right here, that there should be an A in that particular position in the chromosome. But it turns out that it is actually a T. What does that mean? It means that this person is homozygous. Instead of having an A, this person is having a T. Now, this means that it could be a polymorphism, just normal variation, a normal genetic variant, just having, instead of having A, this person is homozygous for that position and having a T. Or it could be a mutation. It could be a mutation that can cause a disease. And we don't know that. We have to confirm it. Okay, so keep that in mind. Now, the thing is, with this Sanger sequencing, the old method, the problem is that it's laborious and it's not safe because we're dealing with radioactivity, and radioactivity can be harmful. Okay, so scientists came up with a better approach, a safer approach, using fluorescence. Fluorescence is not harmful. Now, here, the primer is not really labeled. Rather, it's the dideoxynucleotides. These are the ones that are labeled by fluorescence. So A emits, it gives a color that is red. C is blue. G is green, and T is yellow. So each one of them gives a certain color. Instead of having 4 reactions, we're going to have just one reaction. And this one reaction is going to contain the template, the primer, same thing as the Sanger sequencing, the other sequencing. Well, this is called Sanger sequencing as well because it's based on dideoxynucleotide. So we have the template, we have the primer, we have the enzyme, we have the substrates, regular substrates, deoxynucleotides, deoxy A, deoxy C, deoxy G, deoxy T, and so on. And then the 4 dideoxynucleotides. But each one of them gives a certain fluorescence, okay, as shown in here. Do not memorize, of course. Okay, and then we have DNA synthesis. Remember, again, we have many molecules of templates, many molecules of primers and substrates and enzymes and everything else. So let's say we have 1,000 template molecules. If there's an A and the DNA polymerase synthesizes the template, if there's an A, it's going to add either a deoxy T or a dideoxy T. Okay, remember that here we have small amounts of these dideoxy molecules, so most probably it's going to add a deoxy T. It can add a dideoxy T to that DNA as well, to a small portion of the DNA template. So out of these 1,000 molecules, let's say that 50 of them would terminate at the dideoxy T. Now this molecule that terminates with the dideoxy T is going to give the whole thing, the whole molecule is going to give a signal of a certain fluorescence, right? Because now the dideoxy, the label dideoxynucleotide is part of it. Now let's say that it sees a G. It's going to add a deoxy C or a dideoxy C. Most probably it's going to add a deoxy C because it exists at higher concentration. But for some, let's say 50 or so, it's going to add a dideoxy C. Now, this DNA fragment now that terminates with the dideoxy C is going to give a signal, and the signal is going to be blue, right? But for the other molecules, for the other templates, the synthesis continues, and so on. So each dideoxy gives a certain color. So we're going to have different DNA fragments synthesized. If they are terminated with a dideoxy molecule, then it's going to give that a certain signal. Okay, and these DNA fragments are different from each other by 1 nucleotide. So each one is different than the other one by 1 nucleotide. Now someone says, well, wait a second. What if the DNA polymerase adds deoxy T to all templates? Well, you know what? It's a matter of probability. So it's really statistics, right? So it's a matter of probability. It can add a deoxy T or dideoxy T instead of 50. Let's say it's going to add to 45 or to 55. So there's going to be variation, right? But it's not really all or none. It's not going to be that case. Okay, it's always going to be that the sum of the molecules of the DNA molecules are going to have a dideoxy molecule, dideoxynucleotide. Now, it gets faster because instead of us looking at the X-ray film that we use to look at the radioactive signals, we use, it's automated, it's computerized. So the instrument is connected to a laser, a detector, and the detector is connected to a computer. So the fragments are gonna be separated according to size. So here we have the short fragments and here we have the long fragments. And again, it's read the same exact way, upward. That's the 5' end of the newly synthesized strand. And we're going upward, we're going toward the 3' end. Okay, so we have sample. So instead of having 4 lanes, for each lane represents a dideoxy G or dideoxynucleotide, dideoxy G, dideoxy C, T, and so on. No, rather we put them all in the same lane, and the fragments migrate, and they are separated according to size. And if they differ by 1 nucleotide, then they're going to be separated. And the detector reads the signal of these fragments right here. So it's going to say, I see a red, then, oh, I'm colorblind. I see a blue, a red, and so on. Now, the computer translates the colors that it sees into peaks. It's just, a way of representing the data. And it tells me that there's yellow. Um, Again, I can't see the colors. Whatever that is, blue, green, whatever. Then there's a reds here. Then we have green, and it translates the colors into letters. Okay, so it tells me that the sequence of this DNA molecule is whatever, C, G, A, etc. Okay. Um, Again, the sequence represents the newly synthesized DNA from 5' to 3'. So here we have the 5' end, and here we have the 3' end. Of course, once we know the sequence of the newly synthesized strand, we can flip it and have the sequence of the template molecule. So this is safer. So it's automated. It is safer. It's more joyful because we see colors instead of black and white. So here we have, let's say that we're comparing sequences of 2 individuals. One is healthy and the other one is diseased. So we're looking at a sequence of a certain gene. And we have these peaks, beautiful peaks. Okay. So these peaks are translated into letters. So we have C, T, A, etc. Okay. So that's the normal sequence. Now, someone says, why is it that we have variation in the length of the peaks? Well, because again, it's a matter of probability. You might have 50 DNA strands having a T, then the following one we're going to have 45 or 40, the other one's going to be like 55 or 60. So the intensity of the peaks differs a little bit. Here we have a person having a variation in the sequence. So we have C, T, A, again, C, T, A, C, etc. And then we have 2 peaks that are overlapping. What does it mean that we have 2 peaks? Again, just like the previous slide, it means that the person is heterozygous. We're diploid. 2 chromosomes are synthesized at the same time. C, A, G, and then we have T, and then on the other chromosome we have a T, let's say. So A on one chromosome, T on the other chromosome, so we have 2, and that's why we have 2 peaks migrating, 2 bands migrating at the same time, at the same level, but the detector sees 2 colors, green and red. Aha. And then it tells me that there's a C and an A at that position. So I know that this person is heterozygous. Now, let's say that we have a mutation, that the person is homozygous, having a mutation, and the mutation is homozygous, meaning that it exists on both chromosomes. What do we expect in terms of peaks? Are we going to see a single peak or double peaks? Well, you guessed it right. A single peak, but the color is different. So by comparing the unknown or the DNA of a patient with the healthy DNA or DNA of a healthy person, we're going to see that instead of having a C, we're going to have a T, let's say. So we say that this person is having a mutation, and this mutation exists on both chromosomes, so the person is heterozygous. Sanger sequencing has been powerful, and we still use it till these days, except that it's time consuming, it's laborious, and it's not really cost effective, it's slow. So scientists came up with different approaches that can be faster, can be computerized, integrating bioinformatics, different information technology tools, and so on. And they came up with several technologies. One company called Illumina came up with a nice technology that is quite powerful, quite fast, and cost effective. And instead of taking a short piece of DNA, it actually can sequence the whole genome of an individual. Remember, the Human Genome Project took 15 years, almost 15 years, to complete 90% of the human, to sequence 90% of the human genome, and it cost $3 billion. A lot. Nowadays, we can sequence the genome of an individual in a day, and the cost can be as little as $100. Instead of $3 billion, just $100. The development, the progress in technology, the improvement in technology has been amazing when it comes to DNA sequencing. And this approach, the new approach of sequencing DNA, is known as next generation sequencing. And pay attention to me. So here's the concept. Okay, here's the concept. The next generation sequencing takes advantage of the genome that we already know, the human genome that we already know. We know what the sequence of the human DNA is, and it's freely available online. So you take the DNA of an individual. Again, we draw blood, for example. We extract DNA from saliva, buccal swab from the mouth. We can extract DNA, and remember, we're talking about good quantity of DNA, many molecules of DNA of the double-stranded DNA, of course. So this DNA, once it's isolated, is fragmented. So we have different fragments. Notice that the fragments, by the way, since we have many molecules of DNA and the DNA is fragmented randomly, we're going to have overlapping sequences. So we're going to have, let's say, look at here, for example. Look at this right here. We have 3 fragments right here, and there are overlapping sequences right here and right there. So let's zoom. Can I zoom in? No, I cannot zoom in. Okay. But here's the thing. So we have an overlap between these sequences, right? And between these 2 fragments. Okay, so that's what we have. Then these fragments are taken and DNA adapters are added to them. So these are short DNA fragments that are added at the ends of every single fragment, and we call them adapters. Okay. Then these DNA molecules are added to a solid surface right here. Okay. This solid surface contains DNA fragments as well that are complementary to the adapters. So each one of these DNA molecules is going to bind to a certain DNA sequence. Okay, so we're gonna have this DNA fragment is added to this at this position. This DNA fragment is gonna bind to that position. This DNA fragment is gonna bind to that position. Now, the solid surface contains many DNA molecules, short DNA molecules that are complementary to the adapters. So each DNA fragment is gonna bind at a certain place, and it's random, by the way. Nothing is organized. Then each DNA fragment is amplified, okay, at each position, forming a cluster. So we have a cluster right here that contains 100 DNA fragments. So starting with one, we're gonna amplify them by PCR-like method. So we're gonna have 100 fragments sitting in this place. We have 100 fragments sitting there. So instead of having single fragments, okay, we're gonna have in each cluster, in each position, we're gonna have 100 of the same identical DNA sequences. Wonderful. And then DNA polymerase is added, and special nucleotides are added as well. And DNA synthesis starts, except that the nucleotides that are added, they are not dideoxynucleotides. And they are not normal deoxynucleotides as well. These nucleotides are added. They have two characteristics. One, when they are added, the DNA polymerase cannot add the following nucleotide. It cannot. Okay? Until the nucleotide that is added is modified. Then the DNA polymerase can add the following nucleotide. Okay, so that's one. Two, the nucleotide gets activated and it gives a fluorescence. So the DNA polymerase comes in, it adds a nucleotide. The nucleotide then is activated. It gives a signal of fluorescence. And then it's modified so that the DNA polymerase can add another nucleotide. When it adds this nucleotide, it gives a signal. And then it gets modified so that the DNA polymerase adds the third nucleotide, and so on. So every time the DNA polymerase adds a nucleotide, the nucleotide gives a signal, and then it's modified, and the following nucleotide is added. Okay. So when it gives a signal, now each cluster is different than the other one, right? So each cluster is going to give a specific signal. This cluster gives a red signal, indicating that in this cluster we have a T that is added. A neighboring cluster, there's an A that is added, so it gives a, I don't know, a blue signal. Okay, and then a third cluster that is far away, we have another nucleotide that is added, green fluorescence comes out. Okay, so, and we have a camera on top, and the camera takes a picture of all of the colors that are produced at the same time. So it keeps reading the colors coming out of every cluster. And by the way, each solid surface has millions of clusters. So we have millions of colors coming out at the same time. And the camera takes a picture of all of these colors, all at the same time. Amazing technology. So here's the idea again. So let me repeat it. So here we have the concept again. This is taken from the book. We have DNA. DNA is fragmented randomly, and we have adapters that are added to the ends of every fragment. Then they are added to the solid surface. The solid surface contains millions of probes, millions of DNA fragments that are complementary to the adapters right here. Then, in each cluster, we're not going to go into details. In each cluster, we're going to have amplification of the DNA strand that is bound to that cluster. Okay, so we're going to have hundreds of these DNA fragments of the same sequence in each cluster. Great. Okay, by something like PCR. Don't worry about the details in here. Then we go to the next step. Let's say that we're looking at these two clusters right here, this one right here and this one right there. Okay, so for that one, for this cluster right here, we have the DNA fragment with the adapter, and on the solid surface we have the DNA that is complementary to the adapter bound to that solid surface. So the DNA binds to the DNA fragment on the solid surface, and we add the DNA polymerase and the nucleotide, the special nucleotides. So it sees an A, it adds a T. Now this T has to be modified for two reasons. One, to give a signal, that's one, to emit a signal. Two, it has to be modified so that the following nucleotide is added. So it gives a signal and it's modified so that the following nucleotide is added. Notice that the signal that comes out of this nucleotide is different. The first one was blue, the other one was green, and so on. For that cluster right here, notice that here T is added and then G is added. Now let's look at the other cluster right here. Okay, so it's the same adapter and it binds to the same DNA fragment on the solid surface. We add the DNA polymerase and the nucleotides. They're all added to the same solid surface. Okay, so the DNA polymerase sees a T, it adds an A, and A gives a yellow, colorblind. It gives a yellow color, and then it's modified so that we add the following one, which is AC, which gives a red color, and so on. So this cluster, the sequence of this cluster is a T, then G, then A, then C, and so on. The sequence of that cluster, the other cluster, is an A followed by a C, then another C, then a T, and a T. So the sequence of the DNA fragments in each cluster is different. Again, we have 4 clusters right here. Round 1, we add the DNA polymerase and the nucleotides. So this cluster gives a yellow color. This cluster gives a green color. This cluster gives a blue color, and then a red color. Round 2, the cluster gives a blue color. Then this one gives a green color. This one gives a red color, a yellow color from this one, and so on. Okay, so after each round, each cluster gives its own unique color. So the sequence of the DNA in this cluster right here is G, C, T, G, A. For that cluster, it's a C, then a T, then a T, then an A, then a G, and so on. All right? So, it's really, really fireworks happening really fast. We're talking about enzymatic activity, and we're talking about millions of clusters. And the camera takes a picture of all of these colors, and it can distinguish the different clusters from each other. So it reads the different colors coming out of all clusters. It's like fireworks. Okay, and then what? And then um, the DNA sequences of all of these fragments are taken and they are put together because there are overlapping sequences. So it's gonna piece them together. It's gonna put them together one after the other, and we're gonna have a continuous DNA sequence. So again, you take DNA from yourself, fragmented, many billions of copies of DNA, overlapping sequences of DNA fragments. These fragments are added to the solid surface. They're going to bind, adapters are added. They're going to bind to different places. And then we're going to have synthesis of DNA. Each time the DNA polymerase adds a nucleotide, the nucleotide gives a color at a certain cluster. It gives a color, it's modified, the following one is added, and then gives another color or the same color, whatever. And then the sequences are collected and they are pieced together into something that looks like this. So we have one continuous sequence of DNA. I encourage you to watch this video from the company. Now, this video contains a lot of information. Many of them are not really necessary for you to know, just details, but it gives you an idea of how things work. And I hope you enjoy it. Allah ma'akum.